what we see is that people with the ApoE4 allele, and so increased risk of developing Alzheimer's disease, have lower levels of plasmalogens. People with the ApoE2 allele have higher levels of plasmalogens. So this relationship between the ApoE genetic risk factors and plasmalogens in circulation seem to agree with the concept that plasmalogens, and this is a lifelong exposure because it's a genetic risk factor. So people with a lifelong exposure to low levels of plasmalogens have a higher risk of developing Alzheimer's, and if they have higher levels, they've got a decreased risk of developing Alzheimer's disease. Now, we then went on within this study to do what's called a mediation analysis, and that's where we really asked, is the relationship between the ApoE2 allele, in this case, and the decreased risk of Alzheimer's disease, is that mediated by the plasmalogens in circulation? And what we saw was that, yes, it is mediated by plasmalogens in circulation, and that mediation can explain about 30-35% of the protective effect of the ApoE2 allele.
Hello, Dr. Meikle. You are the head of the Metabolomics Laboratory at the Baker Heart and Diabetes Institute. So welcome to Modern Healthspan, and thank you so much for joining us today.
Thanks, Richard. It's good to be here.
Thank you. So, Dr. Meikle, I think plasmalogens are kind of very important components of the body. However, they have not received a lot of attention so far, especially in kind of the aging area. So can you talk about what are plasmalogens? What are the key functions of them?
Okay, so look, plasmalogens are phospholipids. So there are lots of lipids in our bodies. Cholesterol is a lipid, triglycerides are lipids, and phospholipids are another class of lipids. And phospholipids are particularly important as they're major components of all the membranes around the cells and within the cells of our bodies. And so plasmalogens are one of the types of phospholipids that make up these membranes. Now, the unique properties of plasmalogens have to do with the particular type of linkage. So the phospholipids are made up of a choline backbone, a glycerol backbone, a choline or ethanolamine head group, and then two fatty acids or fatty alcohol groups. And the linkage between one of those fatty alcohol groups onto the glycerol backbone is called the vinyl ether linkage. And so it's different from all the other phospholipids that occur in our bodies. And the unique property of that particular linkage is that it acts as an antioxidant. And so I think this is one of the main properties of plasmalogens, is when they're within the membranes of our bodies, they can protect those membranes against oxidative stress. And of course, that's a key component of many metabolic diseases.
So they're mostly in cell membranes. So what percentage of the cell membrane do they make up?
So that varies depending on the tissue type that we're talking about. So if we think about the brain, then plasmalogens make up more than 50% of the phospholipids in the membranes within our brain. So that's a very high proportion. In heart, they're also very high, about maybe 40%. In immune cells, they're also typically quite high, 20 or 30%, and then other tissues have lower levels. So it really depends on the tissue type and the function. But typically we see that tissues that are metabolically very active tend to have higher levels of plasmalogens.
So this would be like brain cells and heart cells?
Yes,
would they?
Exactly.
Okay. So the plasmalogen has two lipids attached, two fats. So I'll call them fats because they're fat. They're fats, right? Are they?
Yeah, they're essentially fatty acids attached to this glycerol backbone.
How important is it the type of fatty acid? I mean, is it helpful if it's unsaturated or saturated?
So as you say, there are two two fatty acids attached to the glycerol backbone. The one that carries, or the one that's connected by the vinyl ether linkage is typically either a saturated or monounsaturated. So it's a 16-0 or an 18-0, and that encodes 16 carbons but no double bonds, or an 18-1, 18 carbons with one double bond. So that's the alkyl or alkenyl linked fatty acid. The other fatty acid, though, for plasmalogens is typically a polyunsaturated fatty acid. So that might be, DHA, so, the fatty acid that's typically in fish oil, omega-3 type fatty acids, but also might be arachidonic acid, so an omega-6 type fatty acid. And so plasmalogens have a role within the body of also not only, in terms of a structural role within the membranes, but they also have a role in carrying these polyunsaturated fatty acids around the body and then releasing them under certain conditions where those polyunsaturated fatty acids are needed for other processes.
Okay, let's come back to that in a second. So one of the you said that it works as an antioxidant because it has this vinyl link, which has like a double bond. What happens once that has been used, that bond is broken? Does it cease to be a plasmalogen, or does it get rebuilt?
It does. So what happens is that plasmalogen is broken down, and so then the body needs to make more plasmalogens to replace that. The problem occurs if oxidative stress can attack membranes in various ways. So it can hit the plasmalogen, and that breaks down the plasmalogen, but that stops the oxidative stress from propagating through the membrane. If the reactive oxygen species instead attack a polyunsaturated fatty acid on another phospholipid, then it creates a reactive radical on that phospholipid, and then that can then pass on to another phospholipid, another polyunsaturated fatty acid. And so you can get this propagation of oxidative stress moving through the membrane. And so the role of the plasmalogen is to break that propagation, so limit the damage that's done by reactive oxygen species.
If you have sufficient lipid antioxidants like vitamin E, would they, in preference, deal with the reactive oxygen species?
Look, of course, vitamin E is an antioxidant, and there's been lots of studies done by giving vitamin E to protect against oxidative stress, and in some situations that may have some effect, but a lot of the clinical studies to try to use vitamin E as an antioxidant haven't really been that successful. Of course, the antioxidant has to be in the right place at the right time to interact with those reactive oxygen species that are generated within the cells.
You said that plasmalogens, one of the roles they have is to deliver fats to the right location, and particularly like an omega-3 like DHA or something like that. So could you talk a little bit more about that? I mean, I thought the fats were kind of attached to the glycerol backbone, and that's how they stayed. So it takes them somewhere, and then they get split off.
Yeah, so within cells, and so firstly, plasmalogens also circulate within our blood. So lipoprotein particles, LDL and HDL, they have a cholesterol and triglyceride core, but the surface of those lipoprotein particles are made up of phospholipids primarily. And that includes plasmalogens, and so plasmalogens can move around the body. But within the cells, there's a range of different phospholipases. And so phospholipases are enzymes that cleave off the fatty acids from phospholipids. And so if the cell decides that it needs to have free DHA or some other polyunsaturated fatty acids, then it can activate the phospholipases. They can cleave off these polyunsaturated fatty acids from phospholipids and then provide them to the cell to be metabolized into other, typically other signaling molecules.
So where do we get plasmalogens from? Can we get them from the diet, or do we make them inside ourselves?
So both of those answers are correct. So we synthesize plasmalogens ourselves in all the cells in our body. So the plasmalogens are the first part of the synthesis occurs in organelles within cells that are called peroxisomes. So peroxisomes are very important organelles. You have to have peroxisomes to synthesize plasmalogens. There's a number of genetic diseases where people have dysfunctional peroxisomes or they have dysfunctional plasmalogen synthesis. Those are typically very severe genetic diseases, typically impact in early childhood, major neurological symptoms, and a range of other problems with people who have those genetic diseases. So our ability to synthesize plasmalogens is critical. We can also get plasmalogens from the food that we eat. So, but of course, plants typically don't make plasmalogens. So we get plasmalogens from meat, from eggs, from fish to some degree. But, so mainly animal products are sources of plasmalogens for us.
We absorb the plasmalogen complete, or does it get broken down in the gut and then we kind of rebuild it?
Yeah, again, it's another complex story there. So plasmalogens are very sensitive to acid. And so in our stomachs, we have very acidic environments, so very low pH. So certainly some of the plasmalogens are broken down in the gut or in our stomachs at that low pH, but some seem to get through. And so when we look at large population studies where we've done our lipidomic profiling, so we measure all of the lipids within the blood of individuals, including the plasmalogens, and then we've got records of what those people ate, we can see that there's a relationship between people who eat a lot of red meat or a lot of eggs. They have higher levels of plasmalogens circulating in their blood. So that's how we know that at least some of the plasmalogens can get through the stomach and be absorbed and then be, circulate in blood.
I saw that the natural plasmalogens that we use as supplements came from shellfish, but it seems that eggs have more plasmalogens. Is that correct?
Not necessarily more, but certainly from our studies, of course, people don't eat that much shellfish, and in fact, the studies, the dietary information we have on our studies doesn't discriminate between fish and shellfish. So we see, all right, there's a dietary category of fish or seafood, and we see that's also associated with a positive impact on plasmalogens. But of course, people typically eat a lot more red meat, for example, than they would eat shellfish.
Do you know what the percentage is in terms of where the plasmalogens come from, whether it's from diet or endogenous manufacture?
Good question. Good question. Look, I would expect that the bulk would come from endogenous synthesis, but we don't actually have any hard evidence on this, to tell you the truth. Of course, we could calculate how much is in the diet, but then we don't know exactly how much gets through the acidic conditions in the stomach. We don't know how readily that's absorbed. So there's still a lot of things that we don't know about how plasmalogens are taken up from our diet, I think.
Would there be any precursors that would help you to make plasmalogens?
Yes. So look, the field has used Because plasmalogens are quite difficult to synthesize. They're quite difficult to purify from different food sources. And so when people have wanted to study plasmalogen or modulation of plasmalogens, then typically they've gone to a class of compounds called alkyl glycerols. So that's the glycerol backbone that's present in phospholipids with an alkyl chain attached. So that's a fatty acid with an ether linkage. So it doesn't have the vinyl ether linkage, it's just got an ether linkage, but it's part of the way there to being a plasmalogen. Now, those compounds can be ingested. They're quite stable to the acid conditions of the stomach. So they get through the acid conditions of the stomach, they get absorbed into circulation, and they can be metabolized into plasmalogens. Okay, and so if you take alkyl glycerols in your diet, then you can increase your plasmalogen content in circulation and in different tissues.
Are these molecules, are they natural? I mean, we eat them normally, or yeah?
Yes, so alkyl glycerols, there's one major source that's used, and that's shark liver oil. And you can buy shark liver oil capsules. Now they actually contain what's called an alkyl diacyl glycerol. So it's the alkyl glycerol that I just described, and the other two arms, if you like, of the glycerol backbone have fatty acids on them. So it's like a triglyceride. It's like it looks similar to a lot of the fats that we would have in our diet. So that's the natural source. You can chemically synthesize the alkyl glycerol. And so that's also been used in a number of sort of preclinical studies where we want to look at the effect of modulating plasmalogens in different mouse models.
What happens to plasmalogens as we age?
Okay, so again, to answer those questions, we go back to a lot of the large population studies that we've done. So in my lab, we do lipidomic profiling. So we take a small amount of blood or plasma, typically, and we extract all the lipids out of that plasma, and we measure about 800 different lipids in those small samples. And now we can do that on large numbers of people. So anywhere from a few hundred up to 10,000 people in some of our studies. And so if we look at the lipids in circulation across a population, then we can ask ourselves, well, exactly your question, what are the plasmalogens levels in people who are older compared to people who are younger? And what we typically see is plasmalogen levels decrease as we age.
And do we have any idea why?
Why? That, again, good question. There could be, well, and there are likely to be a number of reasons for that. One is as we age, typically levels of oxidative stress might be increasing. And of course, oxidative stress would break down plasmalogens, and so they may be just turned over quicker in different tissues. So that would explain some of the decrease that we see in plasmalogens in older people. But we just might not be as effective at synthesizing plasmalogens as we age. We know that our cells don't perform to the same level of efficiency. And we see that in mitochondrial function, for example. That's the sort of organelles within our cells that produce energy. But I think we also see that in peroxisomes, and these are the organelles within the cells that produce plasmalogens. So if they start to lose function, then the rate that we can produce plasmalogens will also decrease.
Right. And we think we make most of them internally. Does it matter what kind of, I think kind of discuss it, does it matter what kind of fat gets put on the plasmalogen? Because I mean, one thing I'm thinking about is like omega-3s. Omega-3s are required, so we can't make them internally. So if you don't eat them, they definitely can't be put on the plasmalogen. So would that cause any impact?
Yeah, look, there's two sides to that question. There's the alkenyl chain that's attached to the plasmalogen with the vinyl ether. And as I say, typically that's a 16-0, an 18-0, or an 18-1 in humans, at least. Now, what we see when we analyze all the plasmalogens in humans, that the 16-0 makes up about 40 or 50%, the 18-0 maybe 30 or 40%, and the 18-1 makes up only 10 or 20% of the plasmalogens. Okay, and so the challenge then is when we look at something like shark liver oil. Shark liver oil is mostly 18-1 alkyl glycerol, and so is converted into 18-1 plasmalogen. So if you take in a lot of shark liver oil, you increase your plasmalogen levels, but typically you increase the 18-1 type of plasmalogen at the expense of 16-0 and 18-0. And so there's subtle differences, but we don't really understand how big an effect that might have over the long term if you change the composition of the alkenyl chain on the plasmalogen. So that's the one side. The other side is the fatty acid that you mentioned that might be a DHA or a 22,6 fatty acid. Now, obviously, it's important that they are part of the plasmalogen. They're in the 2 position of the glycerol backbone. The thing there is, though, that they can be removed and replaced. So there are enzymes, as I said, phospholipases, that take those fatty acids off the plasmalogen. And then there's acyltransferases that put a new fatty acid onto the plasmalogen. So they're constantly being remodeled. So whether you start with a 22,6 or a 24 or an 18,2 in that position, that can be changed. But the alkenyl chain can't be changed. That's there and it's fixed until the plasmalogen is completely broken down. So there's, again, a lot of complexities in the structure and the number of different types of structures that you can have.
I heard that C16-0 is pro-inflammatory in general as a saturated fat. If it's attached to a plasmalogen in that, does it have the same effect?
No, it doesn't. Firstly, it's not there's a number of reasons why C16, so palmitate, has a pro-inflammatory effect. And again, we don't understand all of them, but one of the pathways is that C16, so palmitate, is one of the basic components of a class of lipids called ceramides. So C16 or palmitate combines with serine, and that's the first step towards the synthesis of ceramides. So if you have a lot of C16, you tend to increase the level of ceramides that your body produces, and ceramides are pro-inflammatory lipids in a number of situations. Of course, the C16 on the plasmalogen or the alkyl group there, again, that can't be released until the plasmalogen is completely broken down. And so it's not contributing to that inflammatory effect. In fact, plasmalogens are typically anti-inflammatory.
Excellent. So what is kind of the outcome of having lower plasmalogens? I mean, as we get older, we have lower plasmalogens, but in disease cases, do we also have lower plasmalogens?
Yeah, again, so we go back to our population studies and we look at, what are the plasmalogen levels in people who have heart disease, for example. And we see that, again, people who have heart disease, or even people who are going to develop heart disease over the next 5 or 10 years, they will typically have lower levels of plasmalogens than people who don't have heart disease or who aren't going to develop heart disease. So that we see that, on multiple studies looking at heart disease. We also see the same effect on Alzheimer's disease. Again, people who either have Alzheimer's or who are going to develop Alzheimer's will have lower levels of plasmalogens than those who aren't going to develop Alzheimer's. Now, of course, there's no cause and effect there. That's an association. So it doesn't mean that plasmalogens will protect against heart disease or Alzheimer's disease, at least not from those observations or those analyses. But we have done a number of other studies and a number of other analyses where we do what's called a mediation analysis. And that gives us more causal inference around the role of plasmalogens in different disease situations.
Okay, can you talk about those in some more detail for these kind of studies?
Yeah. So look, I think that the Alzheimer's is an important one because Alzheimer's disease, is a growing problem in our health systems. We've got an aging population in most countries in the world. And so Alzheimer's is increasing in many of those countries. Now, if we just take a step back, there are environmental risk factors for Alzheimer's disease, and there are genetic risk factors for Alzheimer's disease. One of the major genetic risk factors is a gene called ApoE. And so you can have an ApoE2 allele, or an ApoE3 allele, or an ApoE4 allele. So there are three different variants of this particular this particular mutational variant in the ApoE gene. If you are ApoE4, you have about a 3 to 4-fold increased risk of developing Alzheimer's. If you compare to the E3 allele, if you have an ApoE2 allele, you're about half as likely to develop Alzheimer's, so you have a reduced risk. So what we were able to do in some of our larger studies is say which lipids are associated with the E4 and the E2 alleles. So we look at 1,000 people, some have an E4 allele, and we ask, what are the levels of plasmalogens in people who have the ApoE4 allele? What we see is that people with the ApoE4 allele, and so increased risk of developing Alzheimer's disease, have lower levels of plasmalogens. People with the ApoE2 allele have higher levels of plasmalogens. So this relationship between the ApoE genetic risk factors and plasmalogens in circulation seem to agree with the concept that plasmalogens, and this is a lifelong exposure because it's a genetic risk factor. So people with a lifelong exposure to low levels of plasmalogens have a higher risk of developing Alzheimer's, and if they have higher levels, they've got a decreased risk of developing Alzheimer's disease. Now, we then went on within this study to do what's called a mediation analysis, and that's where we really asked, is the relationship between the ApoE2 allele, in this case, and the decreased risk of Alzheimer's disease, is that mediated by the plasmalogens in circulation? And what we saw was that, yes, it is mediated by plasmalogens in circulation, and that mediation can explain about 30-35% of the protective effect of the ApoE2 allele. Okay, so now we're getting to the point where we're starting to see evidence for a causal role of plasmalogens to protect against, in this case, Alzheimer's disease.
Could you talk a little bit more about the mediation? I'm trying to understand how you determine that the plasmalogens are mediating the difference, and then come up with this 30% number.
It's a complicated statistical analysis. I have to say that we have a group of bioinformaticians in my lab that do all of this analysis, but essentially you can measure a direct association between the ApoE and the outcome, in this case, Alzheimer's disease, and then you can add into that regression model, you can add in the plasmalogens and see whether the plasmalogens offset some of the direct effect, and so you can get an estimate of the mediation effect of the plasmalogens in that situation.
So I think there have been some interventional trials with plasmalogens as well. Have any of these looked at Alzheimer's or other neurodegenerative diseases?
I think there's been a couple where they've used, and I think you might be referring to some of the shellfish plasmalogen type products where they might have looked at cognitive outcomes. I'm not sure about those trials. They're generally small. They're generally using fairly low levels of plasmalogens in the diet in terms of the level of supplementation. So it's hard to see how they've whether I guess it's the validity of those types of studies, I think, relies on being able to reproduce them. And I think we need a number of additional studies where we can reproduce the effects of that type of dietary supplement, particularly in a field as complex as neurodegeneration, where you're measuring cognitive decline and there's a lot of variation in those measurements. So we've still got a long way to go, I think, before we can say, right, yeah, plasmalogen supplementation, that's going to be the solution, or at least part of the solution to Alzheimer's disease or other neurodegenerative diseases. Now, that's not to say that isn't going to, evolve into that story, but it's going to take quite a few much larger studies and more, more controlled studies, I think, to get sufficient evidence to support that.
Okay, so at the moment, what we can say is that it seems to be in some way causal, and it is correlated with Alzheimer's, but we haven't really seen that supplementing with plasmalogens helps helps particularly.
Yeah, look, we need larger studies with hard endpoints, I think, in that space.
Do we see that supplementing with plasmalogens raises the plasmalogen level? And I'm just thinking, so you can measure it in the blood as well. So do we know whether it would raise it in specific tissues?
Yeah, so we've done mouse studies where we've used the alcohol glycerol type compounds, and we feed that to mice, and we see that increases plasmalogens in circulation. It also increases plasmalogens in a number of tissue beds, so in cardiac tissue, in adipose tissue, and a number of others. So yes, we know that you can, at least in a mouse model, you can affect plasmalogen levels in circulation and in different tissues. In humans, the studies are much more limited. We've done one study with shark liver oil in humans. And there we can, again, with a dose that was 4 grams per day of shark liver oil for 30 days, we could see a significant increase in circulating levels of plasmalogens in those individuals. And it was a very small study. It was 10 males that were overweight or obese. But at the same time, we did see that those individuals showed a significant reduction in cholesterol, although that was small, a significant reduction in triglycerides, and a significant reduction in hsCRP, which is an inflammatory marker. So all the signs from that very small study were positive, and so I think we're encouraged to continue down that path.
Plasmalogens, because we talked about AD, Alzheimer's, but they also, as you just kind of alluded to, are related to metabolic and cardiovascular health. So have you done these kind of association studies on the cardiovascular and metabolic side as well?
Yes, so, so in addition to, in the same way that we can see these associations with Alzheimer's disease, we've looked at associations with cardiovascular disease, and that's quite evidence. We've looked at associations with fatty liver disease is another one where we see a strong plasmalogen signature. Again, people with lower levels of plasmalogens have a greater risk of developing fatty liver disease. And we see that both from measuring plasmalogens, but and looking directly at the association with fatty liver disease. But we also see that when we do what's called a genetic correlation. So we use the genetics of the individuals involved to see whether plasmalogens are genetically associated with fatty liver disease. And so again, that's taking out the environmental influences and just looking at the genetic influences, which gives more of a causal inference, although not completely. But we see almost exactly the same profile. People who genetically have low levels of plasmalogens are more likely to have or develop fatty liver disease. So if I can just extend on that, so of course, because we're looking at these different cardiometabolic diseases, and you can see we're from the Baker Heart and Diabetes Institute, that's our sort of bread and butter. Then of course we've then gone on to use mouse models of fatty liver disease or mouse models of atherosclerosis to show that if we supplement with the alcohol glycerols and increase the levels of plasmalogens, that we can actually protect against fatty liver disease, and we can protect against the accumulation of atherosclerotic plaque in these animals.
So in terms of supplementation, I saw that there was kind of natural and synthetic plasmalogens. And does it matter?
I think it does matter. Well, it doesn't matter the source in a sense. What matters is what the actual structures are, I suspect. So as I alluded to before, the alkyl chain or the alkenyl chain of the plasmalogens are typically 16-0, 18-0, or 18-1. And we know that humans have a particular ratio of those different alkenyl chains, and we also know that they can't be remodeled once the plasmalogens are formed. And so our view is that if we're going to supplement plasmalogens, then we should make sure that what we supplement has the right composition, if you like, so the right ratios of those, so that we're not altering the internal ratio of plasmalogens in the cells in our bodies, in the membranes within our bodies.
And is that more likely to be right with natural or synthetic?
Well, I think with synthetic, you can control those.
Right.
You can control what the composition is going to be. With natural plasmalogens or natural precursors of plasmalogens, like the shark liver oil I mentioned earlier, then you don't have control over that. So I suspect that synthetic is going to be the direction that we will need to take, not only because of this compositional issue, but also just from a sustainability issue. So plasmalogens from shark liver oil is possible, but there aren't that many sharks, and there's not that much shark liver oil produced. And so I don't think that in the long term would be a sustainable solution. In fact, so what I do is I take krill oil, and so I didn't mention this. The story starts to become quite complicated. Krill oil also has ether lipids in it. So krill oil is a source of omega-3 fatty acids, but part of those omega-3s in krill oil are attached to phospholipids. And those phospholipids, while not being plasmalogens, are ether lipids and are precursors to plasmalogens. So again, you can take krill oil and you can increase your plasmalogen levels by taking krill oil. You need to take a lot more than is the recommended supplement levels. But if you if you use krill oil instead of fish oil, then you get a little bit of a boost to your ether lipid or plasmalogen content.
Okay.
Again, probably not enough to be physiologically significant, but but there is a little bit of a boost. Although if you're very low, then it might be enough.
It's always common. Well, I eat eggs, so hopefully that helps.
Yeah. Now look at eggs. Eggs are one of the the most abundant sources of plasmalogen, sort of gram for gram.
Right.
And obviously that mostly occurs in the egg yolk. And again, so you can see it. People who eat, several eggs a day will have higher levels than people who eat no eggs a day.
Right. Okay. Oh, that helps.
Of course, just one thing. So we see this eggs and red meat, great for plasmalogens, also great for cholesterol and saturated fats, which are not good for our metabolic health.
Yes.
So there's a trade-off there. Too many eggs and you're just elevating your cholesterol. Too much red meat, you're elevating your saturated fats, which again are not healthy in a balanced diet. So yes, you can use dietary sources to increase plasmalogens, but that needs to be balanced within all of the other components of the diet, which is why I suspect that a diet alone is not going to be sufficient. And that's why there is a need for supplementation.
Yes. One thing I did notice in the interventional trials was when they used natural, they had very low doses, like you said. But when they used synthetic, they had much higher doses, like hundreds of times higher. Do you know why that was? I just wonder.
Now, look, and this is all the issue that concerns me, that there's no reason that you would need less of. If it's the same compound, the same molecule, then there's no reason you would need less of a natural compound than you would of a synthetic compound.
Right.
Having said that, if you're supplementing with plasmalogens themselves, then certainly you will need less, perhaps, because the alkyl glycerols need to be metabolized into plasmalogens, and that process is not 100% efficient. Now, because the alkyl glycerols, or particularly those that you might get from shark liver oil, they look like a fat. Okay, they look like a triglyceride. And so a lot of them will simply get stored in adipose tissue. And others will be broken down through different pathways rather than being converted into plasmalogen. So only, we've done some tracer studies in mice to look at what's the efficiency of conversion from alkyl glycerols or other precursors into plasmalogens. And typically, alkyl glycerol conversion might only be a few percent. So it might only be 3 to 5% from alkyl glycerol that actually gets converted all the way through to plasmalogens. So if you start with the plasmalogen itself, you'll need a lot less than you will need from an alkyl glycerol. You need to deal with the potential losses in the acidic environment in the stomach, but allowing for that, and there are potential ways around that, then you'll get, a higher level of effective plasmalogens from something that's already a plasmalogen than something that needs to be converted into a plasmalogen.
Right. And if this glycerol gets stored in the fat, that's non-metabolic, so it just sits there and just stays there until you burn the fat, in case you do.
Yeah, potentially. And again, it may be useful to have higher levels of these ether-containing triglycerides or ether-containing alkyl diacylglycerols in your adipose tissue, because they will be released over time, and then they may be then converted into plasmalogens at a later date. So again, we haven't really done the studies to identify how long it would stay within the adipose tissue, what happens to it when it would be released, and those types of things.
What kind of dose? So suppose well, that's a good point, actually. So there's plasmalogen supplements that are on the market now, I think, and maybe they have plasmalogens in. If you were using, if you wanted to supplement, would you use like the precursors or the supplement itself? And what kind of dose would make sense for a person?
Okay, so a sort of disclaimer here, if you like. So we're working with a company that is developing a plasmalogen supplement. So we've done a lot, or we've had a lot of consideration about what dose level do you need, depending on what the precursor compound might be, or whether you're dosing with plasmalogens themselves. So I told you earlier about shark liver oil. And when we dosed with 4 grams per day of shark liver oil, alcohol glycerol, that gave us about a 20 or 30% increase in plasmalogen. So that was, that had a biological effect on the men that we dosed over that time. So that's one end of the scale. As you develop precursors that are more bioavailable, and so more of them is converted into plasmalogens, then you might need much smaller doses. So the type of compounds that we're looking at now, we think probably around 200 milligrams per day would be sufficient. If you were using plasmalogens themselves, you might get away with even less than that. You might get away with 50 or 100 milligrams per day. But again, I haven't seen anyone do the studies as to see what portion of those plasmalogens would make it through the acidic environment of the stomach. So that would need to be overcome, I think, for if you were dosing with directly dosing with plasmalogens.
You should be able, I think, to put the plasmalogens inside what they call an enteric capsule or something, that it would get it through the stomach, but it still needs to get past the lipases in.
No, I think that side of things should be fine. We know that the lipases in the stomach and intestine, they'll cleave off the fatty acid from the SN2 position, but the alkenyl chain is stable to lipases. Okay, and so what you'll have is a lysoplasmalogen will get absorbed, and then, as I said, that can be reacylated to form a full plasmalogen.
So let's go with shark liver at the moment. So, because that's something that exists right now, right? Do plasmalogens have like a half-life? I mean, if you take like rapamycin, right, it stays around, but then it's got half of it's gone in a day or so. Is it the same kind of thing for plasmalogens, or they, once they're there, they get embedded in the cell wall and they just kind of stay?
No, well, I mean, lipids and phospholipids are sort of quite active, so they're turned over on a regular basis, and it depends on a number of things. It depends on the type of phospholipid. It depends on the environment that they're in, how quickly they will be turned over. So in some situations, plasmalogens might have quite a short half-life in the order of hours. In others, they might have quite a long half-life in the order of days. So it really, and of course, then, we've talked about adipose tissue that where it might be even longer than that. So it really depends on the situation that the plasmalogens are in as to how rapidly they might be turned over.
If you were low on plasmalogens, which would imply that across your body you have low levels in various cell walls, would it take some time to get to a steady state, even if you're supplementing? And I mean, is there any way we can estimate how long that would be?
We've done this in mouse studies. So if we feed mice a supplement of alcohol glycerol, and then we monitor them over a period of days or weeks, we see that typically the level of plasmalogens in circulation will increase, and it maybe takes a week or two until it reaches a plateau, and then it will be relatively stable at that new level within those animals. So yes, it does take time. It's in the order of days or maybe a couple of weeks to increase. When we look in some of the tissues, it might be a bit slower, so it takes longer to get incorporated into the tissue, so maybe 3 or 4 weeks to reach the plateau. But typically in that order of time.
Is there any way for consumers to measure or estimate how they're doing in terms of plasmalogens?
Look, not really, not at the moment. We're working on that also. So we have, we do lipidomics and we measure plasmalogens in blood from thousands of people. But we do this as part of these large clinical studies or or population cohort studies. We haven't established that as a commercial service at this point in time, although we do have plans to do that. Because the other thing that we are interested in using our lipidomic technology for is to measure risk for different diseases. So we can measure risk for cardiovascular disease, and we can improve on the current risk score. So at the moment, people will have their cholesterol measured, their LDL and HDL cholesterol. The doctor will also assess whether they're diabetic, what their blood pressure is, whether they smoke, and a number of other risk factors. And from that, they'll calculate a cardiovascular risk. Okay, what we can do is we can measure several hundred individual lipid species, and we can recalculate or re-stratify people into higher or lower risk groups. And we find that's more accurate than the existing tests. So we're in the process of translating that into a clinical test. And so at the same time, of course, we're measuring a number of plasmalogens. And so and in fact, plasmalogens form part of the cardiovascular risk panel or the risk profile. But at the same time, we can just extract out the information just on plasmalogens. And so we can assess whether someone has high levels of plasmalogens or low levels. And so we hope to have a test like that available perhaps in the next 12 to 24 months.
Okay. And then you would be able to measure the plasmalogens, but in the end, it's true that the important number is actually your risk factor. Although I guess identifying whether plasmalogens are the largest part of that risk, that your low plasmalogens would be very important so that you could know what action to take.
Yeah, look, and obviously as more supplements come on the market, then I think, people would want to assess, do they need to take a plasmalogen supplement? If they already have high levels because of their diet and their genetic background, then perhaps they don't need to take a supplement. And again, but we don't quite appreciate, as you increase your plasmalogens within the normal range of human physiology, we see a decrease that's associated with the decrease in a number of metabolic and neurological diseases. But if we push the level of plasmalogens higher than the normal range, do we see an even bigger decrease in risk? And again, we haven't done those types of studies. So the question would be, what is the appropriate level? And that might be different for different individuals.
How good is measuring plasmalogens in the blood in terms of a proxy for what the measures are in the various tissues? Does it correlate directly?
Yeah, look, we've done a little bit of that. There is a correlation, and again, depends on the tissue. So I think if we look at, for example, plasmalogens in the blood and immune cells that are, exposed to the blood to a large extent, then there's a correlation there. If we look in other tissues, although we have limited data, I think, available in other tissues to really assess that accurately. But I think what we do know is that if you increase, or if you take a supplement, at least in mouse models, to increase the level of plasmalogens, then it does get into the tissues as well as in circulation.
Including
so
into
know that. Sorry, go ahead.
So including into the brain?
Look, this is a much more challenging area, I think. Of course, the brain is protected because there's the blood-brain barrier. And so whether plasmalogens will cross the blood-brain barrier is a little bit unknown. It depends on again, probably depends to some extent on the type of precursor compound that you're using. We know that there are transporters in the blood-brain barrier that transport lysophospholipids typically. So these are phospholipids that have one of the fatty acids removed. And so it may be that lysoplasmalogens could more readily cross the blood-brain barrier than plasmalogens themselves. But also, we know that there's a relationship between circulating plasmalogens and Alzheimer's disease. And so why is that? One is that, circulating plasmalogens are perhaps reflecting what's occurring in the brain. But of course, there are also cells that cross the blood-brain barrier, and typically some immune cell subsets can cross the blood-brain barrier. And so it may be that, you're changing the composition of those immune cells that get into the brain. And so that has an impact on inflammation and oxidative stress within the brain, which are drivers of Alzheimer's disease. It may be that plasmalogens influence the blood-brain barrier itself. And we know that in Alzheimer's disease and other neurological diseases, there's often an increased leakiness in the blood-brain barrier that contribute, or is believed to contribute, to disease processes. And so whether plasmalogens are involved in that side of things remains to be seen. So, there's a number of issues in relation to increasing plasmalogens in the brain, or treating or protecting against Alzheimer's disease or other forms of dementia, that aren't necessarily clear cut. And it's not necessarily about, we have to increase plasmalogens in the brain. Overall, it might be a particular cell type within the brain that we need to target.
But we don't really know at the moment. What we do know is that if you have higher blood plasmalogens, that is associated with lower rate Alzheimer's.
That's where we are at the moment. And I think there's a lot of biology, there's a lot of clinical testing needs to be done to understand what the best strategy is and what will be most effective in protection against Alzheimer's disease in particular. I think it's a challenging area.
What are the next steps for plasmalogens in your lab? What are you looking at?
So we're looking at a number of preclinical models, and I mentioned, we're looking at cardiovascular disease, we're looking at fatty liver disease, we're looking at the role of plasmalogens in immune modulation, and so potentially plasmalogens also have a role in terms of how our immune system develops or responds to different stimuli, such as vaccines. And so those preclinical studies are going on at the moment. We're working with a company to develop a supplement that will be suitable then for humans. And that will then also allow us to have enough material so that we can do human clinical trials in some of these areas. And so I think over the next 2-3 years, we'll be looking to start some of these clinical trials specifically to assess, for example, can we use a plasmalogen supplement to improve someone's response to a vaccine? And this is obviously an issue, particularly in older people or people with comorbidities that often respond poorly to a vaccine, such as a COVID vaccine or an influenza vaccine. can we boost their response to those vaccines and so give them better protection? Can we, reduce the risk of people developing fatty liver disease in that space? And then at the same time, these are all clinical applications, but at the same time, there is the potential as a dietary supplement just to boost your plasmalogens and reduce your risk overall of cardiometabolic disease and potentially other neurological diseases.
But plasmalogens are are part of the cell wall, right? So they're functioning to make the cell wall have certain features. These, like improving the immune system, that works through improving the cell walls of the immune cells. It's not doing signaling in some other way.
Look, there are multiple roles for plasmalogens. As I say, they do contain omega-3 and omega-6 fatty acids, and they're signaling molecules when they're released from the plasmalogens. But look, one of the main roles of plasmalogens is to protect the membranes from oxidative stress, and that's a process that occurs in immune cells. There's been a number of studies now to demonstrate that plasmalogens can protect against that oxidative stress, and in immune cells there's a particular process called ferroptosis where you get a high level of oxidation on the cell membrane, and that ultimately causes cell death. So some of our studies show that plasmalogens can protect against that process. There are other studies, of course, that actually indicate that plasmalogens may enhance that process, presumably because plasmalogens are carrying the polyunsaturated fatty acids that are also oxidized as part of that process. So there's still a little bit of uncertainty there, although I suspect, again, it depends on the particular situation and the particular cell type that you're looking at. Our experience is that plasmalogens will be protective in that situation. So they'll protect immune cells from ferroptosis, from that oxidative cell death that occurs in a number of stress situations.
For plasmalogens, is there anything else that we should discuss?
Look, the one other thing, just quickly, early life development. This is it's relevant because early life development is when our immune system is developing. It's when our brain is developing, and we've done a number of studies now looking at infants and their mothers, and looking again at their lipidome in circulation, and looking at the relationship between breastfeeding and the level of plasmalogens in the circulation of the infants. And what we see is infants who are breastfed have higher levels of plasmalogens than infants who aren't breastfed. We also see that those infants have lower levels of inflammation and lower levels of infection over the first year of life. Now, when we go back and we look at the breast milk itself and compare that to infant formula, we see breast milk has more than 10 times the level of plasmalogens than infant formula has. So this is an important, we're seeing the same effect. We're seeing plasmalogens protect or help to modulate immune development and provide some protection to the infants in the early stages of life against infection and inflammation. And they're derived completely naturally, of course, from the breast milk.
That's interesting. So right from the start, like the plasmalogen level.
From early life to the more or less the end of life, I would say plasmalogens are going to be important contributors to our health.
Cool. So on that note, where can people go to kind of follow your work and keep up to date?
They can go to the Baker website. And, my lab has a web portal there where we put up, a lot of our technology and methods and so on. But also we have a list of our publications up on the lab portal and on the Baker website, so they can follow the publications that we're making each year.
The company that you're working with, is it still in is it stealth mode? Does it have a website?
It does. So there's a parent company called Juvenescence. And so you can look on that. And then there's a daughter company called Pelagic AI, is the daughter company that's sort of started up specifically to focus on building the pathway for plasmalogen supplementation, and they have another product in development as well.
Okay, Dr. Meikle, thank you so much for joining us.
No problem, Richard. My pleasure.